Welcome back to my channel. Today we discuss about the process of transcription. What is transcription? We discuss in this video. Okay, some friends commented me that in night time some other sound are irritating them. It is the last concept I study in night, and the further classes I take in the daytime. Okay, it is the last class in night time. The process of transcription we discuss briefly in this class. What is transcription? The process of converting genetic information from DNA to RNA is called as transcription. The process of information of DNA sequence to RNA sequence also is called as transcription. By the process of transcription, the RNA sequence converted from the DNA. Okay. When a complete DNA sequence is present, at the time a RNA polymerase is joined at a part. After joining a RNA polymerase, the two parts of the DNA are get attracted. When get attracted each other, it separated from one to another. The lagging strand separated from this leading. The leading strand separated from this lagging strand when RNA polymerase is joined at this region. When the process is start, then the complete sequence converted into three parts. One is promoter, the middle part is regulator, and the last part is terminator. The work of the promoter region is it starting the process. Promoter promote the process, and terminator end this process, and regulator regulate the whole process. Okay, when promoter promote this transcription process, at the middle the regulatory region is present. Regulatory region make a RNA copies as compared to the leading strand. That means when a RNA copies make, the complete sequence is changes. If in the leading strand, if the lagging strand is A, T, G, C, then when a RNA copies is made, the opposite strand is present. That means A oppositely present T, but here T is not present. U is present because thymine is replaced by uracil when a RNA copies is made, so that the process is transcribed as changing by RNA model. A opposite to A make U, T make A, G, C, G. A completely opposite DNA, opposite sequence of RNA is made, and it is continue when the process is not reaching at the terminator region. When the process is reaching at the terminator region, then the process is end, and a completely sequence of RNA is formed. We know that RNA is a single-stranded protein sequence. It is starting from 5' to 3', we saw in this picture. That means a completely RNA sequence is made from the from one DNA sequence. Okay. When a complete RNA sequence is present, here some coding and some non-coding region is present. The exons part is called as coding sequence, and the intern parts is called as non-coding sequence. We remove the non-coding sequence by the process of speciosome. By the process of specialism, we remove the non-coding sequence or intern sequence from this RNA part, RNA sequence, complete RNA sequence. After removing the non-coding sequence, a completely mRNA is present or converted from this making RNA sequence that is transcribed from the DNA sequence. Okay. After that, the whole information of DNA is converted into the RNA. Okay. Here the sequence is present A, U, G, C. Okay. After getting a complete DNA sequence, it gets out from the nucleus and come into the cytoplasm. That means I tell you in a video that in a cell inside the nucleus DNA is present, but RNA present in the cytoplasm. When a complete RNA sequence is transcribed by the process of transcription, then it comes out from the nucleus to the cytoplasm. This is about the whole process of transcription. Thank you all of you to see my video, and all please subscribe, like, share with your friends. Thank you all of you.